What you need to tell me is if my screen is still visible, it should be.
It is. It is.
All right. So this is a lecture. This series of 2-3 lectures or 4 lectures. Sometimes it expands a little bit. Is on the general principles of gene regulation and gene regulatory networks as they arise. Now, to really understand what gene regulatory networks are and the complexity of it, we have to first understand the fundamental unit that gives the gene regulatory network a subunit or a unit that can be used repeatedly by the cell itself to construct these networks. Okay, and that fundamental unit is called an operon. Okay, and we will discuss those with 3 examples, one of which you have already seen a little bit, and the other 2 we'll talk about. We mentioned it, but we have not discussed it in detail. Okay. So what I've given you as an example here is what we call a transcription network, and I've picked this up from a slide that we'll use in a future lecture. But the reason I'm putting it here, so you see the connection when this one you see again in the future lecture, which I will talk about when we talk about transcription networks. Now, what you see here is an example of how signals can affect genes. What we have shown here in this figure is that there are n number of these signals, which go through some transcription factors, which are m in number, because there could be some common transcription factors. And then you have genes which are being activated, or sometimes it could be also inhibited also. In this case, we have shown all the arrows to be activating, and they go from A to K and K in number. So this itself lays a framework, if you wanted to develop a mathematical model, because now you know there are n signals, m transcription factors, and K genes that are getting activated. So that is the sequence in which you will look at things, right? So this lays down that fact, but also what you notice is that one signal could affect multiple genes through maybe 1 or 2 transcription factors, or you can have multiple signals required to activate one gene, which is the second example, signal 2 and 3, and so on. So these different kinds of motives that are there, they come under the study of what are called network motives, which we'll again do in a separate study, so in a separate set of lectures. But to get to all of those things, we have to start with the fundamental unit that is called the operon. Okay, so how does this work? Before we get to that point, let us just look at the principle of how this transcription occurs. So essentially, you'll have one signal which will give you this control of transcription that takes place. That means there will be a position in the gene where you will have RNA polymerase come in, and then that will start synthesizing the RNA. And as the RNA gets synthesized, you'll get the ribosome coming in, and that will translate and give you the protein. Okay, and the protein will again have post-translation modifications if that is required, and otherwise it will become either active or inactive depending upon whether it got folded correctly or not get folded correctly. So large proteins usually have a problem of folding, and you have what are called chaperones that help it to fold if they are misfolded. And otherwise also, these proteins which are folded or misfolded or are present in the cells will over a period of time get degraded and broken down to the fundamental amino acids that are made up of that make up these proteins, and those will then go back and again get reincorporated in some new proteins that are getting synthesized. So you have got what is called the transcription level, at which your main primary code is getting converted to an mRNA, and then you have a translation level where you have the code which is now in an RNA form gets translated to what is called a protein. So that is the way this whole thing works. But this regulation that takes place here, that's one level of regulation, and the other level of regulation is at this point. Now together this might be part of what is called a regulon, which could an operon, and these operons can then have multiple of these operons can together be part of what is called a regulon. So the regulon is the superset of operons, and operon is is going to, and one operon is going to be a fundamental unit from which gene regulation is understood. Clear? Okay. What I'm going to use here is one example of such a very well-known operon, and this is the lac operon. So how many of you, I mean, I think all of you who are from the biochemical department should be able to understand this, have come across this. But what about others from other departments? Have you had any background? If you have taken SBL100, you should have seen this. If not, let me know. Yeah, Malik, you might be muted, I don't know.
No, sir, nothing. No problem, sir.
You don't know anything about this, is it? All right. So look at the operon itself. First of all, when you have to do an analysis on the operon, this itself shows the level of complexity, because what you have got is a site for the lac inhibitor I, a protein that is there. This is the regulator protein, and that has a separate promoter site, which is separate from the main section. Here, for this sequence where you are producing three proteins, one is beta galactosidase, the other one is a pyrimidase, and the third one is a thioglycosidase. So that's also a protein which is what called a trans-acetylase, which is shown in green here. So green, purple, and the blue here. Okay, and they're normally named as LAC-Z, LAC-Y, and LAC-A. Okay, so these are the three main proteins. These are going to be the structural proteins. Sorry, structural genes which are going to produce the proteins, which are going to be acting in some way to kind of help the cell to utilize some components that are required, right? All right. Now it has a promoter site and an operator site, right? So now the RNA polymerase which is there to synthesize this has to come and sit ahead of the on the promoter site, and then from there it has to synthesize this. But you also have what are called the operator sites, which are essentially places from which the regulation takes place. Now, when this protein is getting synthesized, this is an active regulator for repression. So I think we should not use the word active, because we just say regulator for repression, because there are others which are activators. So, and then active and activator gets confusing. So just regulator protein, and it's a repressor in this case. And in the absence of this regulator protein, the transcription can proceed, right? So whereas, if this binds here, then it binds in such a way that the operator that binds to the operator in such a way that it does not allow the RNA polymerase to come and sit on the promoter site from which it can start transcribing. Okay. Now, what happens there? There are 2-3 things that are there. It looks very simple, but I'll just first talk about one part of it, and then we'll talk about the other part. The first part of it is very simple. It just is based upon negative control. What is the negative control? The negative control is this, that if there is, so normally this protein will be, synthesis of these of these structural genes will be not operational. That means it's not going to do this in normal circumstances. So normally what happens is, especially in the E. coli and the bacteria, they would prefer to have other sources of carbon, preferably glucose, to be utilized, preferred over other carbon sources for cell energy, right? Now here what happens is, if you have no glucose present but you have lactose present, that's when this starts working. So normally, to keep this lactose utilization blocked down, this inhibitor in the presence of glucose will actually be present, and it will be blocking this site, and you will require some kind of a signaling molecule, and in this case an allolactose, to come and remove this from the site so that this transition can take place. Now, How does this allolactose happen? Actually, there is some kind of a basal level of production of beta-galactosidase, which is normally present. And so when there is, the presence of, whenever there is lactose present, there is this allolactose that is getting synthesized by this beta-galactosidase, and then that goes and binds to this repressor, removes it, and then allows the synthesis, which essentially tells it that where there is lactose present, but there's no glucose here, and so therefore this lactose has to be utilized, and so it starts synthesizing this gene. Right? Clear up to this point?
Yes, sir.
Yes. Okay. Now so I need to ask you several questions here. So how many of you would like to would understand that, or can appreciate rather, that there is this timing that is required, right? There is a timing that is important. So what kind of times are important here? One time would be, let's say, the presence of lactose and realization by the cell through this circuitry that lactose is present. And the other is the utilization, the synthesis of these proteins so that the lactose can be utilized for energy, right?
Yes, sir.
So keep that in mind. So now if you were writing a model, for example, as in system value we do, if you try to explain this this operon in terms of some mathematical equations, then you have to keep in mind that the time constants are going to be different, right? And that will actually essentially, we'll see later on also how that becomes very important, because there are these circuitries which are very well timed. So especially when you're looking at a regulon, where you have multiple of these operons present inside this regulon, then how and what sequence of this synthesis takes becomes important. And this goes back to the first few lectures we had, where Ratnesh and we were having a discussion about the creation of an artificial cell, where this complexity is so huge that is still not clear how we can actually utilize that. Clear? That's one of the big bottlenecks that we have. Yeah. All right. Now, there is another part of this complexity. So this is the simplest part of it, right? So this is just the inhibitory control, and it tells you that, well, there's a negative control that is present. Now, it so happens that actually glucose along with CRP AMP, that means cyclic AMP receptor protein along with AMP synthesis, these two together actually exercises a positive control on the same lac operon. So you have two kinds of control here. Keep that in mind. And this is somewhere where people get a little confused. So they kind of work independent of each other, but the effect is combined, right? So now let's look at this independently first. If you look at this independently first, what it is telling you is that in the presence of low glucose, usually there will be higher amount of cyclic AMP present. And this goes to another set of regulation which we're not talking about. The reason is because one reason is, if you look at the PTS system, to get the glucose into the system, you require ATP. When there's excess of ATP present, there's another signaling that takes place which converts this ATP into cyclic AMP. So when you have low glucose, you have high cyclic AMP. So that's the way the cell is balanced always. And usually if you look at secondary metabolism taking place, I don't know how many of you have actually looked at it, there is a cyclic AMP signal that takes place. So when the trigger for a secondary metabolism to get initiated is the end of the utilization of the carbon source and the cyclic AMP spike that takes place, which is commensurate with that, and maybe an oxygen signal that is present. So if you're modeling secondary metabolism, then those things become important. But here we are looking at only glucose, CRP, and cyclic AMP being a positive regulator for this same lac operon. How does it work? Well, there is a cyclic AMP binding site near the promoter region, and CRP binding site with the cyclic AMP bound to it. So when there is excess of cyclic AMP, then there is a modification that takes place in this. There's a modification that takes place of this protein, and together they can actually bind. If one of them is not present, it will not bind. So once this is bound here, it essentially enhances the rate at which the RNA polymerase can bind here by about 50-fold. So that by itself is, in the presence of lactose, can convert, can give you conversion of, in the whether lactose is present here or not is independent, but essentially it will be able to convert the lactose because the downstream processing takes place here for the proteins that are going to be synthesized from the Y, Z, Y, and A genes of this operon, right? Now, the other counterbalance of this is that when you have high glucose and low cyclic AMP, even if lactose is present, the amount of the upregulation that takes place here is not as much. Okay, so the binding will be fewer. The reason why it's shown empty is because this is all based on frequency. So here the higher frequency of binding will show as if most of the time it is bound there, but here because of lower frequency of binding, most of the time it will appear as if it's empty. So although it is empty here and the lac is bound here, there will be some leaky synthesis that takes place. So always, although this is quite an elaborate binding that takes place on 2-3 promoter regions of, there are 3 promoter regions really for this depressor, and 3 sites, not promoter, 3 sites on this sequence where O1, O2, and O3, where it can actually loop around. In spite of that, there is some leaky synthesis that takes place. So there are 2 things that are happening therefore. You're getting a positive control by glucose, CRP, and cyclic AMP together. And on the other hand, you have the lac repressor protein, lac-I, which can get released by allolactose or its homolog, like we have an IPTG, which is used in the labs. And that can actually release that binding protein, and then you get the synthesis, right? Now, together what happens? Together what happens is this, that essentially if you have this promoter site released, that means there is no lac repressor bound because there's a lot of lactose present, and you have also low glucose and cyclic AMP present, then you have a very high rate at which the synthesis takes place and the lactose is getting utilized. Whereas if you have this not bound, but you have high glucose and low cyclic AMP, still you get some of this synthesis taking place, some rate at which this gets synthesized and you get utilization, but it's at a much lower rate. And you can get the worst case situation so you'll have 4 cases, right, that takes place. And the worst case situation will be when you have this high as well as this is bound and there's no cyclic AMP, then you'll have the least amount of this when you have no lactose and when you have this bound, plus you have high glucose, then that's when it's nearly shut off. Okay. Any questions on this so far?
No, sir, it's pretty clear.
Yeah. What about the others? I'm a little concerned because this is a very fundamental thing that others have to also understand from other departments. And if we need some assistance here, this is a point that you have to ask, because after this, we'll not come back to this. Okay. There is another kind of control that is there. This is called the positive control. Okay. So what we saw was a negative control with the inhibitor of the lac operon by the lac-I protein. And here what we're looking at is another very classical example, which is called the arabinose operon. And you have what is called, and this also requires, the synthesis of 3 proteins, BAD, Arabad, which is along this side, which are the structural proteins. And those can then be used to utilize arabinose. Now, here again, we have, this entire DNA that is there, this part of it. Essentially, so this is going to be get locked into place when you have, there are 3 sites here. So you can see one is an ARA-O2, which is an operator site, and the ARA-I1, which is the inhibitor site, where this ARA-C protein, which is there, which is synthesized independently, will actually come and bind here. And this loop that forms here will essentially block the synthesis. But you also see there's a cap. This also the CRP and cap are usually used interchangeably. So it's again the cyclic AMP receptor protein. It's the same thing, but it's also called cap protein. Now, here when you have arabinose present in the system, then the binding is such that it essentially will immediately trigger the synthesis. Normally, that's what happens. So it's not going to. So this is an activator that essentially will allow the synthesis of binding of the RNA polymerase and the synthesis of the structural genes straight away. So one situation is that you have like a valve. If it is normally closed, then it's an inhibitor protein I, like I protein. That is called a negative control. And if it's normally open, that means normally the synthesis is taking place, then it's called a positive control, and it will promote the activation of the RNA polymerase and the synthesis of the proteins that are required for synthesis. So as soon as arabinose is present and the cyclic AMP is present, then this will get activated. It's very simple as that. Okay, so this is the signaling molecule which immediately starts forth, go ahead and synthesize the Arabat genes which are there, and you get the synthesis of this protein that are required to utilize arabinose. Okay, all right. So now this is a very fundamental diagram of negative control and positive control, or regulation and positive regulation. So how do you understand this? The way to understand this and keep in to remember this is that one is an inhibitor, right? So the inhibitor is either pre-bound and you have to release it, or it is bound to the site along with another molecule. Another molecule is required to help it to bind to that site for inhibition, or the inhibitor site, and that has to be released to release the inhibition. But the primary action of this is that it will actually inhibit. Okay, so the whatever protein sits here will inhibit, but either it has to be released by a binding or it has to attach by a binding. So that is the difference that is there. And here you have positive regulation. So either normally it will bind there and you get the synthesis as it is, and to release it you'll have to make it negative, you have to have a signaling molecule that binds to it and releases it from the site, or you have it is bound there because there is a signaling molecule, and when the signaling molecule is removed, it releases it, and so there is no longer synthesis that takes place. So the action is that if it is bound, then it's going to allow the synthesis, and if it is unbound, then the synthesis will not happen because it requires that conformational change so that it can start bind to that location and then start synthesizing. Clear?
Yes, sir.
And this is so we had seen two kinds, negative, positive, and this is attenuation. And we had seen this earlier also in a previous lecture, but I'll go through this quickly so that we can kind of revise what we understood in the context of gene regulation, the fundamental units of gene regulation. Here, it is neither on nor off. It is a situation where you have a graded response to the amount of a certain signal being present. In this case, if you are synthesizing an amino acid, then the amount of amino acid that is present in the system will give a graded response to how much of this gene is getting translated. Okay, and so there is this repressor, which is that TRP repressor, which is independently synthesized. Then if there's tryptophan that is present, then that allows it to bind to the operator site. And if it binds to the operator site, then it starts a sequence of events. But that sequence of events is through a leader sequence. Leader sequence means a prior sequence before you see the main structural genes that are there. And there is within this leader sequence, there's what is called an attenuer sequence. And this sequence is predominantly used so that it can actually control whether or not this RNA polymerase go further down and permit the synthesis of these genes. Okay. All right. Now, this is a very elaborate slide, but the reason I'm giving it to you here is more for an information rather than for you to understand the background. The information is that there are these 5 structural genes A, B, C, D, reversely named, numbered here, and as you can see here that you will require all of these together to work so that you can convert coordismate to tryptophan. And so you have at low levels of tryptophan, you essentially are able to synthesize the entire sequence. And at high levels of tryptophan, you are only going up to this point where it essentially disengages and you get a stoppage of the synthesis further from here, this point. Okay. All right. Now, as I told you before, these are the different sequences here. So So the sequences are showing you the main components of this leader and the leader side, which is there. So you have within it regions of 1, 2, 3, and 4. Okay, and this is up to this sequence. After this, you have the site of transcription taking place, and from this onwards, it starts synthesizing. Now so from here, you have to keep in mind that 2 and 3 are complementary, 3 and 4 are complementary also. Okay, so that means that you can have a bonding between 2 and 3, or you can have bonding between 3 and 4, and they will give you different structures. And these structures, when they are formed, if 2 and 3 are formed, then they don't inhibit the synthesis of the RNA sequences. But if 3 and 4 are formed, then it inhibits the synthesis and gives you a stop sequence, which essentially removes the ribosome from that sequence and stops the sequence of the further translation of the sequence that takes place. Okay, so this is the DNA, and that is the leader on the DNA. So usually what will happen is the RNA will be sitting on it and sequencing this out to give you the RNA polymerase, which is shown here, right? So this is a little bit ahead of the ribosome, this polymerase. So, and these are close together, of course, as we've seen it earlier in the one of the previous slides. So if you have a presence of excess of at any of the tryptophan, then there's one kind of synthesis that takes place. That means essentially it can complete the sequence here. The leader sequence has got 2 of these trip codons, and this is used for most of the amino acids really. So they'll have the trip codons or the codons for those amino acids in the leader sequence, so that if the amino acid is not there in sufficient quantity, then the tRNA for those amino acids will stall, because it is waiting for this amino acids to come so that it can bind to that site, and then it can go forward. But if there's sufficient amino acid present, then essentially it will synthesize fast and it goes over this sequence and then it goes further, right? So that's the way it happens. Oh, okay. So let's look at what really happens here. So when there is when there is a high amount of tryptophan present, essentially it'll when there's low amount of, so this is actually when there is low amount of tryptophan present, right? So when there's low amount of tryptophan present, it should essentially stall here and allow this 3 and 4 to synthesize. If 3 and 4 is being synthesized by the mRNA, they essentially come together and it forms a terminator-like sequence, and this terminator will essentially stop this synthesis taking place, right? If there's high amount of so whenever there is high amount of this, so this is high amount of ribosome. Sorry, when there's high amount of when there's high amount of tryptophan present. Sorry, I made a mistake there. When there's high amount of tryptophan present, it essentially will go over this and when there's high amount of tryptophan present, it essentially will go over this 2 and allow this, so that 2 and 3 cannot form, and 3 and 4 will form. And because 3 and 4 forms, it will essentially the ribosome will kick out when it comes to this point, because this is a stop sequence. Okay. All right. So next slide shows you when you have low tryptophan present. If you have low tryptophan present, then essentially it's going to stop here and wait. Once it waits here for this tryptophan to come, 2 and 3 essentially will bind, and 2 and 3, when it binds, it is not a stop sequence, so that when this condition is fulfilled, this tryptophan will move forward and then it will pass through this 2 and 3 and then continue sequencing and give you further the entire sequence is then produced. All right. Any questions up to this point?
No, sir, it's clear.
Okay. Sorry. So anyway, sorry about the disturbance that happens. People just. All right. Now this essentially tells you the life cycle the protein that is getting synthesized, how it happens. So this is the sequence of the levels of information that are there. The first level of information is essentially the gene. The second level of information is at the level of the RNA. The third level of information is the level of the amino acids, and the fourth level is at the level of the metabolites that are being present here. So level 1, level 2, level 3, level 4. So essentially these levels are all interlinked. There is, if nothing else, there is a degradation signal that goes and how it's supposed to degrade. You also can have feedback. So from every level, you can have a feedback going to every other level. For example, you can have a metabolite that can actually go back and inhibit, right, a protein, or can go and repress a certain synthesis of a certain, or the translation of a transcription of a certain gene, right? So these things can happen. So if you were to look at these as layers, then just in this sequence from DNA going to proteins and the action of the proteins themselves, which is part of enzyme kinetics, you can understand very clearly that from the enzyme kinetics to the DNA, you are actually able to see 3-4 layers of modeling that can take place, right? Then the question is in system biology is where will you draw the line, right? So on that, let me stop here and let us have a discussion on this so that students who are, or if you have questions, we can go back and discuss this. And there is a little bit of understanding that is required because the next part of this links it to a little bit of the system biology thing that we will be doing later on. Okay, all right. So tell me, let me come back here. So the question now is that I have to ask you, and you need to kind of see if you can interact with me on this, is that where do you think, or what would you think would be deciding at the degree or the level or the detail in which you will be doing a modeling if you were to do modeling of an operon? Just one level. Yes? Somebody can initiate a discussion.
So could you elaborate on that question a bit?
Yeah, so the question that I'm asking here is this, that we are essentially doing system biology, and to understand system biology from a perspective of as a tool to be used for understanding these systems, we need to get into details, right, of how we model this up. Now, if I were to model, for example, all the from a gene getting transcribed and then translated and then they are protein forming and then looking at metabolites. If I wanted to write a model, should I be going into all the details? If I were to, first question is, if I were to design an operon or write a mathematical model for an operon, should I be looking at all the details that are there when I'm writing out these equations? Let's say for the lac operon, take an example.
So, but for the lac operon, since so basically you're talking about some operon that we do not know the characteristics of, right?
Yeah.
So probably we can first we should look at what exactly, first we should look at if it's an inducible or a repressible operon. So if it's a catabolic operon, so it should probably be inducible, and if it's an anabolic operon, it should probably be repressible.
All right, no, all that is fine. So, but the question is, do I include all the detail? Let's say for the lac operon, should I look at the dynamics? Yeah, sorry.
If we look at the key steps where the activation or inhibition is actually occurring, that should do it, and maybe we don't require all the details.
Okay, so that's one.
Sir, we might divide into subparts like as it has been shown in the diagram. So till the generation of from genes to the mature RNA, then to the protein production, that subdivide and then incorporate those status.
Yeah. So actually, it's more like a philosophy question. The reason I asked you is because it should set you thinking. And it's to do with how much detail you want to get into the model and what is the purpose of the model. So if the purpose of my model was to understand, for example, if a mutation in, let's say, a LAC-I protein kind of a situation where you have an inhibitor molecule, and that inhibitor molecule is if the mutation of that inhibitor molecule is something that is important, then perhaps I would have to look at the details of how that inhibitor molecule is getting synthesized, or how that protein is getting synthesized, and those dynamics become important, right? For example, if in a disease model, and I know that, well, there is this one particular gene, let's say angiogenin for ALS, or let's say A beta protein for Alzheimer's, or something like that. So we can choose different protein, or SOD1 for some kind of a coordinated effort with some other protein synthesis, right? So then in that case, we'll have to look at the detail of what is happening at the level of the kinetics involved for an operator binding or inhibitor binding, and those things have to become important, and also the rate of synthesis and how they're getting synthesized. But if I just wanted to look at the operator and wanted to build a unit for an operator, general unit for an operator, which I'm going to use in a regulon, then I'm going to kill all this, not going to be interested in all these details, because here I know that I'm going to look at maybe a signal that comes at the level of the regulon, and the outcome will be maybe at the level of the protein somewhere, and so the smaller details that are there are now going to be important. So it is very important to make that judgment call, and that judgment call can be only made by the person who's doing the experiment, be it although it's a theoretical experiment or a computational experiment. It can only be judged by this person, because the moment you start sitting down with it, it'll immediately come to you, oh, if I'm doing this, I'm getting too bogged down with so many details. It's not really required. Just like you were saying, like you were just saying for just like you mentioned, both Ashi and Ishantan, right? You were just mentioning that you don't need it. So it's quite correct. I mean, but the correctness is dependent upon what is it that you are actually going about to decide in terms of the definition of the problem, right? So the definition of the problem is very important, and sometimes it is also required that that you might have to break it up into layers, because you want to have the freedom that, because it's a computational model, you could switch off. You could design the model in such a way that you could switch off these kinetics that are not required when you're looking at maybe a macroscopic level thing that is happening, as opposed to maybe looking at details of that. When you require it, and so you want to keep that option open. So you can have. So that's where you have multi-layered modeling that takes place. So you have models that are, so it's very important to design the model correctly also, and in terms of the structure of the model, because you should be able to kind of have the flexibility of making the model work for when you're looking at an overall picture, as well as making it work when you're looking at a smaller detail. Okay, so if you're doing a bigger exercise, then those things have to be kept in mind, and these are not always obvious from what your general reading of literature would be, because people don't talk about these things. Clear?
Yes, sir.
Yes,
okay.
sir.
Okay, so
Yes,
I'll stop here today. If you have any questions, please ask now. Okay, then. Sunil, you wanted to say something? If you're saying something, then you're muted actually. Can't hear you. Okay, then I'll leave for today, and thank you for your time.
Thank you, sir.
Thank you, sir.
So thank you, sir.
Bye.